genes, they actually transcribe into different kind of phenotypes. Yes, expression, they are expressed in different ways.
Yeah.
Correct. So genes, they are expressed, either they are expressed in phenotypically in which form, like enzymes are formed, or some visible traits are there, like for example skin color or eye color and all. Yes.
Yeah.
So similarly, once we are talking about the gene expression, so there is a concept of gene regulation also. How we regulate the expression of a gene. Okay, so there is a very complex kind of feature or a process. Correct. Now look here. For example, in our normal body cell, we have all genes. We have all genome. Correct. A normal within one cell of our body, somatic cell, it contains all the genes, but some genes are turned on and other genes, they are, all other genes are turned off. Like for example, I have skin genes. Yes, a skin cell. So in skin cell, only the genes which are related to the function of the skin, they will be turned on. All other genes, they are turned off, despite of the fact all other genes are present. Yes. So in a skin cell, only those genes will be active which are required for that particular function. This is known as gene regulation. This is the simplest explanation that I can give you.
Okay.
So once we are talking about differentiation, yes, so we start our life with a single zygote, and then there are millions of cells, and after that differentiation happen, and each and every cell is doing its own function. So it is regulated. So there are different kind of regulatory mechanisms in gene expression. Any problem?
No.
Okay. Now, obviously, for the gene expression, the basic thing is this is messenger RNA. So, okay, that is messenger RNA. I got it while you're laughing. So, yes. So this is so messenger RNA which was made. Okay. For example, as you said, sir, I'm aware of the fact like what is messenger RNA. For example, this is a messenger RNA. It was the messenger RNA which is made from the DNA. Okay, that messenger RNA is actually referred as pre-messenger RNA. You know this concept, yes? Pre-messenger RNA. You know this concept of introns and exons also?
Yeah.
What are exons? Exons are the genes or the nucleotide, the sequence section of DNA which code for particular kind of proteins, and introns are which are non-coding genes. Correct?
Yeah.
They don't code for anything.
Yeah.
Correct. So once this is a pre-messenger RNA, it is containing both introns as well as exons.
Yeah, but then
so then it is going through a process of spliceosome, DNA splicing or RNA splicing. Yes.
Yeah.
So introns are removed from the exons.
Yeah.
Yes or no? So now this concept, for example, this is exon and this is exon. Okay, this region is exon. So what will be done with this region? These two regions, they will be removed. So remaining DNA, this RNA will be a smaller one. It is made up of a complete
exons
only, and this is a functional RNA. Yes, now it is a functional RNA. Any problem?
No.
Okay. Now one more thing is there. For example, if I divide this thing into, like, this is one exon, okay, this second exon, this is first exon, this is second exon, this is third exon, and this is fourth exon, and this is 5 exon. Yes, 1 2 3 4 5. So I have, how many exons are there? 5 exons are there. So from one single pre-messenger RNA, you can make multiple connections of a RNA. Like it can be 1, 3, 2, 4, 5. Correct?
Yeah.
It can be 1, 2, 3, 4, 5. It can be 1, 5, 4, 3, blah, blah. You understand? Yes?
Yeah.
So with these introns, you can make from a single messenger RNA, you can make multiple kind of products, expression.
Yeah.
This is known as gene expression using the genes itself. Okay, this is one thing. Do you understand this part?
Yeah.
Okay. Now there is a concept which is known as Hussain. So this is again, this is known as pre-messenger. This is once we are after spliceosome, we are getting a mature RNA, messenger RNA, correct?
Yeah.
We are getting a mature messenger RNA.
But this is transcription, right? Or is this gene regulation or transcription?
No, this is gene regulation itself.
Oh.
We will be going into that because, you know, I'm just giving you the concept, what exactly is behind this, how the gene expression is controlled, regulated.
Okay.
Okay. Now, gene expression can be regulated by, you know, there are, have you heard about this thing like a transcriptional factor?
No.
Okay, there is a transcription factor like gene regulation can be made or gene expression can be controlled by transcriptional factor. What is a transcriptional factor? Transcriptional factor actually, you know what I mean? I have to go into detail. Look here, this is the DNA, okay?
Yeah.
In this DNA, and there is what is a transcription factor? Transcription factor is a small section of is a protein which attach to a specific site of DNA. For example, there is a protein. For example, this is a protein, okay? It will attach to a specific site of DNA. This is DNA down. Down is the DNA, okay?
Yeah.
This is DNA. And what is a transcriptional factor? Transcriptional factor is a protein, a small section of protein, which attaches to a specific site or which binds to a specific site of DNA from where the messenger RNA is made.
Okay.
Do you understand this part?
Yeah. Isn't that RNA polymerase?
No. RNA polymerase will come, look here. For example, RNA polymerase will attach here to this transcriptional factor. So transcriptional factor, actually it facilitates the attachment of RNA polymerase.
So it's the RNA.
Or you can, okay, one more thing you can say. For example, if you can say one in another way, you can say, for example, transcriptional factor is attached with RNA polymerase itself. Yes?
Yeah, it's the RNA primer, like this.
Yes,
one.
you can say, correct, yes, this is a primer. Okay, like you can say like this itself, okay? So once we are saying this is the transcriptional factor, yes, it enables the attachment of RNA polymerase to the DNA and it facilitates the production of messenger RNA, correct?
Yeah.
Okay, now this is, now where this transcriptional factor will attach, there are two regions that we call it. One is known as promoter region, promoter sequence, promoter sequence. One is promoter sequence. So once this transcriptional factor, which will attach with the promoter sequence, so it will stimulate the production of messenger RNA. So once the it will stimulate the production of messenger RNA, what exactly it means, or it facilitates the production of messenger RNA. What does it mean? It will increase the gene expression.
Increase.
It will improve the gene expression, correct?
Okay.
No, this is very important. So once, so this promoter sequence is where?
On a
it
specific
is on the DNA.
of the DNA.
Correct? promoter sequence is present on the DNA. So once this transcriptional factor will attach to the promoter sequence, so what will happen? It will stimulate the production of messenger RNA because it is facilitating the attachment of RNA polymerase, correct?
Yeah.
So gene expression is stimulated. One thing. Second word that we use, we call it enhancer sequence. Enhancer sequence. This enhancer sequence is also there on DNA itself. Okay, so transcriptional factor will attach to the enhancer sequence of DNA.
So transcription factor will attach to enhancer sequence and
sequence of DNA. So it will, once the transcriptional factor binds to the enhancer sequence, so it will make some changes in the DNA structure. This is very important thing. So either the DNA structure will open up or it will get compact. This is very important thing which I told. Either the DNA structure will open up or it will get more compact. So if it opens up, it will facilitate the production of transcription, messenger RNA. So gene expression will be stimulated. You got it?
Yeah.
And if the DNA changes in such a way that this DNA becomes closed, packed, it means the gene is actually, or DNA is turned off. It is no more available for transcription. So transcription cannot take place. So it is inhibiting transcription. So gene expression is also inhibited. You got this concept?
Yeah.
Okay, I'm moving next. This is the basic thing. So first, we are using the transcriptional factor for the gene expression. This thing is done now. Okay.
Wait, sir? You said transcription factor attaches to the promoter sequence and the enhancer sequence.
Yes.
So
promoter sequence and enhancer sequence is present with DNA itself.
Oh, so it attaches to the enhancer sequence, and if it like opens up, if it modifies to open up, that's where it attaches to the promoter sequence that makes the RNA.
No. In the promoter sequence, once the transcription factor attaches, so it actually stimulates. So there are different transcriptional factors. Some transcriptional factor, they attach to the promoter sequence. Okay.
Oh,
so once the transcription factor attached to a promoter sequence, it will stimulate.
Yeah.
And if there is another of sequences are there which are known as enhancer sequence. Okay. If the transcription factor attaches to the enhancer sequence, so it will modify the structure, changes in the structure of DNA.
Okay.
So if the DNA structure become, it change in such a way, sometime it will stimulate, sometime it will block. Okay. It will inactivate, it will turn off the gene or DNA. So in this way, gene expression can be controlled by a transcriptional factor. You got my point? So this is done at the gene level.
Okay.
DNA level. This control is taking place at DNA level. We are controlling this transcript, all the phenotypic expressions through genes by blocking or stimulating the gene itself by using transcriptional factor. Is that clear now?
Yeah.
Okay. Okay. Now I'm moving to the next then. Okay. So once we are saying, oh, you are already, there is another concept which is known as epigenetics, isn't it?
Yeah.
What is epigenetics?
No clue.
Huh?
No idea.
So actually, in epigenetics, what you are not dealing with gene, you are not dealing with the a sequence of genes, you are dealing with RNA now, messenger RNA. Instead of dealing with at gene level, okay, with the DNA, like we were doing transcriptional factor, yes?
Yeah.
So even gene expression can be controlled at post transcription also. At gene level, you are controlling transcription, correct?
Yeah.
At gene level, at DNA level, you are controlling a transcription process by using transcriptional factor. Yes. So you are increasing or decreasing genetic or gene expression by regulating the genes at DNA level by using transcriptional factor. Second way of controlling regulating gene expression is by controlling the messenger RNA itself. As I told you, messenger RNA, once it is, for example, transcription has taken place, okay?
Yeah.
Now, transcription, once it will take place, it will make the messenger RNA, that pre-messenger RNA,
Yeah.
correct? So you could not do anything in during the transcription. The gene was expressed quickly.
Yeah.
Before I go into this detail, look here, I will give you a simple example and then I will move on, okay? I think this class will be sufficient for us to, because I don't have to go for another details related to transcription and translation, yes?
Yeah.
So we will try to wind up all the things within this session itself, okay? If even we have to extend the session for 10, 15 minutes, we'll do it, okay?
Okay.
Without charges.
Wow. Thank you.
So yeah, so and sometime it may happen vice versa also. So sometime we will finish before 1 hour, so it will be done. So now it is my turn, so sometime it can be your turn also, yes? So
Yeah.
it is both ways, yes? So the point that I was telling, look, once we are talking about controlling the gene expression using the controlling the DNA, yes?
Yeah.
Expression of the DNA. You know, there are red blood cells and neuron cells. Okay, both of them they have the genes for hemoglobin. Okay, I'm just giving you an example.
Yeah.
Neuron cells also they have the genes of hemoglobin, and red blood cells also have the genes of hemoglobin. Correct? At genetic level. But once it comes to the expression in the cells, so some in red blood cells, you will find out hemoglobin is there. But in neuron cells, hemoglobin, despite of the fact the gene for the hemoglobin are present, but they are not expressed in the yeah, in the phenotype. So you will never find hemoglobin in the structure of a neuron cell, but you will find the hemoglobin in the structure of a red blood cell. So that is what this is known as gene control. So they are regulating by, so for example, I just give you the example, enhancer sequence was there, yes? So in red blood cells, the enhancer sequence, once the promoter transcriptional factor attached to the enhancer sequence, so it opened up the genes which were responsible to make hemoglobin protein.
Yeah.
So transcription took place. But in the case of neuron cells, the transcriptional factor attached to the hemoglobin genes in such a way, okay, the enhancer sequence in such a way, those genes which were responsible to produce hemoglobin, they were blocked. So they were not expressed in phenotypically. You understand? So gene expression can be controlled at DNA level, and it can be controlled at post-transcriptional level also. This post-transcriptional, after the transcription, once we are controlling the gene expression, this is known as epigenetics.
Okay.
If you got this concept, the biggest thing we have covered.
Yeah.
So what is epigenetics? This is controlling the gene expression post-transcriptional.
Wow.
Post-transcription. Mean transcription, look here, we could not control anything. Mean this transcription has taken, this is messenger RNA, this was made, this is pre-messenger RNA, pre-mRNA, yes?
Yeah.
So we could not control, we didn't control anything till the production of messenger RNA, correct?
Yeah.
Now, epigenetics happen after the production of messenger RNA. Then you know the concept already. I taught you about introns and exons, how the exons will be removed.
So post-transcription happens after the mature RNA is produced.
Yeah, you can say like once the messenger RNA is made, that messenger RNA is further, it can be modified. So this messenger RNA, maybe it is available for translation or it is not available for translation.
Okay.
You know, protein synthesis happens in two stages. One is transcription and one is translation, correct?
Yes.
Since you told me, sir, I know it.
Yes.
So what is transcription? Transcription is the conversion of DNA into messenger RNA. This is transcription.
Yes.
And what is translation? Translation is the conversion of messenger RNA into protein. They are expressed in, this is protein, so mean it is phenotypically expressed. Any enzyme is made,
Yeah.
correct? So remember, so what is epigenetics? In epigenetics, we control the gene expression by controlling messenger RNA. For example, this is the messenger RNA, and we want to block one protein. We don't want it. So that particular protein, either it will be stimulated, the production will be stimulated, or production will be controlled, but it will be stopped. You understood this thing?
Yeah.
What does it mean? In look here, in post transcription we are using messenger RNA.
Yeah.
In the other gene expression, in the before transcriptional factor, we turn on or turn off the genes. You understand this concept?
Yeah.
So in before transcription, we are turning on or turning off genes to get transcription.
Yes.
So that was one level. So once the transcription has happened, even at the second level, also you have the body has cell have the control. So at the second stage also they can control if they want, okay, this should be more and this should be less.
Okay.
Despite of the factor the transcription has happened.
Okay, so in transcription modifications for gene expression, the genes are turning off and on, and the genes can also be modified post-transcription, where the genes can either be stimulated or stopped.
No. In post transcription, not genes.
Right.
Post transcription, the messenger RNA. This is known as epigenetics. What is epi means upper?
Yeah, protein.
On. After epi, you can say on top of genes. Now you are not changing anything with the genes. You are not turning on or turning off genes like we were using transcriptional factor to turn on or turn off. Turn on means stimulation and turn off mean inhibition.
Yeah.
So here it is epigenetics, mean on top of genes. So what does it mean? Now we have nothing to do with changing genes. We are dealing with messenger RNA.
Okay.
Got this concept now clearly?
Yeah.
Okay. That is the only thing, you know, in this whole class, if you got this concept, rest of the thing is actually really very easy. Okay. So I'm moving now. Look here, we have this pre-messenger RNA. It has introns and exons. I told you already that how can we remove introns and exons. Introns from one gene, and then it will be a proper functional messenger RNA will get. Yes, mature messenger RNA will get.
Yeah.
And those exons which are remaining, like I gave you the sequence of 1, 2, 3, 4, 5, and then you can rearrange the same messenger RNA can be translated into different kind of products.
Yeah.
You understand? Yes. So if it 1, 2, 3, 4, 5, it will give one product. Okay. But if these exons are 1, 4, 3, 5, 2 like this, so there is another product. So you can reshuffle the same information of the messenger RNA, you can get different products. So this is post transcription changes.
Okay.
Gene expression. So they can be controlled.
Yeah.
Correct? Okay. Next thing is, for example, this is a mature messenger RNA. Okay, this mature messenger RNA, how we can control the gene expression by controlling this messenger RNA now. In epigenetics, we are talking, yes?
Yes.
So here you can say epigenetics, we can do it by doing, there is one term that is known as DNA methylation.
Is it in the paper?
DNA methylation. You know what DNA methylation is there, then histone modification is, it is there, I think in your paper it is there. Just wait, I will show you.
Okay.
So since your teacher have not taught it, so that is why you have confusion.
Yeah.
You know, because normally you are already promoter sequence upstream of the, look here, that is what, whatever we are talking, it is already there. Look here, I'm talking, oh, are you watching my this one? Yes. So you know, I just want to make sure that what we are studying, you have confidence that we are studying the correct thing. Okay. This one you see now, I'm showing you this paper. You can see now. So here if you see promoter dean sequence upstream transcription start and where, okay.
Yeah.
Epigenetic changes, histone, this is histone by modification to the DNA. Epigenetic changes, it can happen, or gene expression can be controlled by modification of DNA that I'm talking now, or modification of the histone.
Okay.
So that is what I was talking now. And afterwards, I will start talking about Lac Operon kind of this thing.
Okay.
Okay. So that is why this is difficult and complex process that we are discussing today. Okay. So it has many dimensions actually. Anyway, I'm just going through with the same stuff again. Okay. I will be using my own explanation. Okay. So here, coming back to this point. So how we can control the gene expression by epigenetics. So there is one concept which is known as, in epigenetics, we can control DNA methylation. You are not changing anything in the DNA, okay? But you put a brake on DNA, that is known as, by DNA methylation, you turn off a gene. What is DNA methylation? You add a methyl group in DNA, and now DNA cannot be used for transcription. So technically, the gene is turned off. Any problem?
Yeah.
So here, for example, if I take this part, this is the most important thing, Tia, okay? This thing you have to make sure like you understand. For example, this is the gene, okay? DNA is there, okay? So once DNA methylation, so in methylation, what we will add? We will add methyl group CH3, yes?
Yeah.
This methyl group, where it will be added? It will be added always, it will be added near to C, cytosine and G, wherever cytosine is there, followed by guanine.
Okay.
Wherever cytosine and guanine are there, this methyl group can add only and only there. Like you remember in the protein structure, the disulfide linkage is always made between cysteines, two cysteines. If you can remember, maybe I did this thing with you or one other kid. Protein structure.
Yeah.
In protein structure, you know, the disulfide linkage is made, okay? So that is made between cysteine and wherever two cysteines are there, they will be attached with each other by disulfide bond, okay? So similarly, methylation will happen wherever C and G will be there, this methyl group will add there.
Yeah.
So once this methyl group will add there, Now this gene is no more available for transcription. So what does actually it mean? It is known as gene got turned off. The genes are turned off now. So no transcription can happen. So this gene cannot be expressed. So for example, in a cell, liver cell, yes, liver cell is responsible, for example, liver cell is responsible to produce bile, yes?
Yeah.
So only genes for the biles, they will, bile producing gene will remain turned on. Other genes which are not relevant, which are irrelevant, all of them, they will get turned off. And how this turn off will happen? DNA methylation is one of the concept.
Yeah.
So DNA methylation can be used to control the gene expression. And demethylation, the opposite one. So if demethylation will happen, then what will happen?
Turning on.
Turning on, obviously the vice versa, correct? Then is the second concept of controlling the gene expression is, the second concept is histone modification. Histone modification. What is the meaning of histone modification? Histone already you know, yes?
The protein around the DNA?
Yes, not around the DNA, under the DNA actually.
Oh, okay.
We both are jingling with the words actually. You know, this histone will be inside and DNA, or it will be wrapped around it.
Okay.
You got my point?
Yeah.
So technically, but actually you were having this thing, you were having this meaning in your mind that what is histone, correct? So if the histone and DNA, okay, the helix combined together, they are known as chromatin. So what is chromatin? A mixture of DNA and histone or chromosome itself. This is chromatin.
Okay.
And if the chromatin is densely condensed,
like the X,
highly condensed,
yeah,
supercoiling, that will be known as heterochromatin. Please, this is very important term that I'm using now, okay? What is chromatin?
It's like the jumbled, like not
a combination, yeah, combination of histone and DNA. Together, it is known as chromatin.
Yeah.
A chromosome with a full chromosome along with this DNA and histones is known as chromatin.
Yeah.
Okay. Heterochromatin, what? Once this supercoiling of chromatin material, supercoiling. So once the supercoiling of the chromatin material will happen, what is the disadvantage? The disadvantage is most of the DNA is not available for transcription because RNA polymerase cannot attach because it is compact. They are not open, they are compact, they are closed. You understand?
Yeah.
So if we are controlling the histone, so we can control the turning on or turning off of gene. So like it was one concept was DNA methylation, adding methyl group, yes?
Yeah.
So now by modifying histone also you can control the gene expression. How? There is one main acetylation. Histone acetylation. One concept is histone acetylation and other is histone methylation. So here histone acetylation you will say, okay, acetylation. What is histone addition of acetyl group?
Not sure.
Okay. Sorry?
Oh, I don't know.
No. Acetyl group, CH3CO, COO, acetic acid, acetyl group. CH3 is methyl group, yes? So acetyl group.
Okay.
I don't have to go into chemistry then, okay? So please remember, acetylation means addition of acetyl group in histone, structure of the histone. So once you add acetyl with histone, so histone, they loosen up. Once they loosen up, it facilitates the gene expression. Why? Because the chromatin material become loose. So most of the genes are available for gene expression, for RNA polymerase to attach and transcription can happen, correct? So it promote gene expression. You understand?
Yeah.
And methylation, obviously, what will be this?
Stopping.
Yes. So adding promote gene expression, So you can refer to the video later on, okay? And then methylation, obviously, methylation, histone methylation. So it actually turn off the gene. What? How? Because the histone, they become really condensed. Due to the methylation, the structure of the chromatin becomes more compact. So if once it becomes more compact, it is no more available for transcription. Genes are not exposed for the transcription. They are not open. So it actually, technically, it turns off the gene.
Okay.
Any problem?
No.
Okay. And so in this way, histone modification and all, we have done this part.
Yeah.
Okay. Now there comes the last thing, which is there in your stuff that we were supposed to discuss, which is really very important, that Lac Operon concept. Okay. Wait, I will show that thing first to you. Okay.
Yeah.
Then
I know it's there. Yeah.
yeah. So here I will be, you have already shared me your picture, yes, for the Operon and all, yes?
Yeah.
Just 1 minute. So can you see my screen and I'm showing your picture here?
Yeah.
Not your picture, but picture of your WhatsApp group.
Yeah.
WhatsApp picture, yes. So if we see it here, no, wait just 1 minute. I will remove it. Yes. Just 1 minute, okay? I have some difficulty because I cannot move. Oh, yes, just 1 minute. Yeah. Okay, now epigenetics, we are done, okay? Now we are on this concept. We are on this concept of Lac Operon.
Okay.
So once we are talking about Operon, do you have any idea what is Operon?
No. No.
An operon is a functional unit of DNA. Functional unit of DNA. Okay? Which contains a cluster of genes. They work for one particular function. For example, again, I repeat, this is very, very important thing that I'm going to discuss with you, okay?
Okay.
Again, what is the operon?
Functional unit of DNA that has a cluster of genes that are related to the same function.
Yes, related to the same function, or they are related to the same promoter. So to the same promoter or same function. So they are actually, their function is one. So for example, Lac Operon, it is actually a cluster of DNA or genes which are controlling the breakdown of lactose sugar.
Okay. Wait.
Lac Operon means operon which controls the metabolism of lactose sugar. So there are three genes available. 3 genes are required which make 3 different enzymes, and those 3 enzymes are required to convert lactose to break down lactose. So some people, they have lactose intolerance, they cannot digest.
Yeah.
Even babies, they are having lactose in, so we have to give them lactose free milk. There are some babies even, And sometime what happened in adults also, in the beginning, they were not having lactose intolerance, but with the passage of time, with aging or something happened, so they become lactose intolerance develop. They cannot digest milk or dairy products. Okay, so why? So this happened because of change in the gene. Okay, now I will be using your the diagram that you have shown me. So I will show you this concept, how does exactly it work. Okay, just 1 minute. Okay, where is your yes, just 1 minute. Okay, the other picture is here. Yeah, now look at this picture now. Yes. Now, if you look at this picture, so one or two things you must remember. Once we are talking in terms of operon, okay, once we are talking, so for example, these are the 3 genes which are responsible for the breakdown of lactose, okay? This will make one messenger RNA, this will make one messenger RNA, this will make one messenger RNA. This will make one enzyme, this will make second enzyme, this will make third enzyme. And all these 3 enzymes are required. These 3 enzymes are required to break down lactose down. Okay. Do you understand this part?
Yes.
Okay. Now, before going into that detail, so you have to remember there is one thing which is known as promoter region, P region. What is a promoter region?
I don't know.
The promoter region which is present on DNA, and on the promoter region, it facilitates the process of attachment of RNA polymerase. Here,
Oh, yeah.
RNA polymerase. Okay.
Okay. What? Okay.
So I will be using one more picture. I think it will give you more kind of, so we will be using this picture also. I will try to explain this thing because we started, oh yes, we are almost on time, yes. So we are not lagging on behind with time. We have 10-15 minutes, so we will finish this thing, okay? Are you okay now, comfortable? Yes?
Yeah.
Okay, now please remember, first of all, the P is standing for promoter region.
Okay.
There is one I is there, which is known as regulatory gene. Regulatory gene. Okay, I is a regulatory gene. Okay, I is regulator. Okay, and then there is O zone is there. O region. This is promoter region. At promoter region, what is the at promoter region, RNA polymerase will attach.
Yeah.
Okay, operational is what? Once it will attach, and technically RNA polymerase has to attach here. Okay, please remember. Okay, and then RNA polymerase, once it will attach, it will start to transcribe this information. This information will be transcribed in this way. This will be this way. This will messenger RNA. This messenger RNA. This messenger RNA will be made. So operator is what? This is the region where the control is there. Operator, we call it operator. Okay, operator region, it is present right after what? The promoter region, P zone. So first is P zone, next is O zone, okay, O region, and third one is actually these are cluster of gene, cluster of gene which are working for one function. What was that one function? To break down lactose.
Yeah.
So these 3 genes, they produce 3 different enzymes, and these enzymes are used up to break down lactose. Any problem?
No.
Okay. Now you know the P zone or P region, you know the meaning of operator. Okay. What is the meaning of operator? This is the zone from where the RNA polymerase will start working. Remember?
Yeah.
Okay. So operator region is present right after P region, promoter zone.
Yeah.
Okay. This picture is not that much clear. So that is why I'm just trying to explain it. I will be sharing my picture also. Okay. So it will be totally clear for you.
Okay.
Okay. Now, then there is another gene which is known as regulatory gene.
Yeah.
What is regulatory gene? It produces a suppressor gene, or sometimes we call it suppressor. It produces suppressor, okay? Suppressor messenger RNA first. From gene, messenger RNA will be made, and from the messenger RNA of suppressor compound will be made, correct?
Yeah.
A suppressor will be made. This suppressor, once it is made, it will attach at operator.
Oh, repressor.
Yes, not suppressor, repressor actually. Pardon with my word, okay? This is actually not suppressor. Okay, technically the word meaning is the same, but we have to use the same word which is there in the book. So it is suppressor or repressor, yes?
Yeah.
Today with one kid, I was talking, like he was saying, sir, DNA duplicates. I told him DNA duplicate, correct, but we have to use a scientific term. It is not DNA duplicate. We have to say DNA replicate, although meanings are the same. But
Oh.
duplicate is English language and replicate is biological term, correct? So suppressor is English term, yes. Repressor is the same, but we will be using repressor instead of suppressor, correct? So this is repressor, yes?
Yeah.
This repressor will go and attach with the operator gene or operator, correct? Or operator zone. So once it will attach to the operator zone, this RNA polymerase cannot do the replication process or this transcription process. It blocks. Any problem?
No.
Okay. For example, this is a repressor. This is the repressor product. Yes, any kind of repressor or cap. Okay. Now I make this repressor in different form. Maybe I'm just trying to copy the same form, but obviously my drawing skills they will. Okay. For example, this is the same. Okay. This is repressor. So this repressor will go here. It will block the operator. So these three enzymes, they cannot be made, which are used to break down lactose.
That's why you're lactose intolerant.
Yes, those people, they have this problem, correct?
Yeah.
Now, sometime what happened, so when this condition will remain, this repressor will remain always here until now I'm talking about a normal situation, okay, normal situation, okay? What is that normal situation? For example, I don't have lactose intolerance. Maybe you don't also have the lactose intolerance, yes?
Yeah.
So this repressor gene is always there on this operator. It is always there.
Yeah.
It will be removed only and only once there is a presence of lactose sugar. Once the presence of lactose sugar will be there, then there will be another gene, there is inducer. We call it inducer, okay? Inducer. It will be produced, one chemical will come here and it will actually what it will do, it will block it. It will attach to this way in such a manner here. It will be blocking it here. Okay. Now it cannot lock this thing any further. So what does it mean? This operator is free now. Nothing is here.
Okay.
You got it? Why? What has happened? This lactose has taken this, look here, this lactose has taken this cap out, correct? It is removed. Presence of lactose only, correct?
Yeah.
So once a new cap cannot reach here, why? Because there was one particular structure, inducer was there. it actually blocks, you know, that enzyme inhibitors, competitive and non-competitive inhibitors,
Yeah.
that concept.
Oh, okay.
Competitive and non-competitive inhibitors. So non-competitive inhibitor, what they do, they attach to the enzyme in such a way that active sites are no more compatible with the binding site of the substrate. So
Yeah.
they cannot. So enzyme is useless now. Same is the case here. This repressor is changed by this particular product in such a way, by inducer in such a way that it can no longer attack and attach here. So what does it mean? This operator zone is, or the operator gene is free.
Yeah.
Free means this RNA polymerase will come and it will start the process of transcription. These genes, they will be transcribed to this. One, these genes will then from this messenger RNA, these enzyme will be translated, correct? And these enzyme will be used to break down these sugars, lactose sugar. So in this way, this Lac Operon concept works. Any problem?
No.
So I don't think that I have to show you my picture that I wanted to show. The thing already you got it now.
You can share a picture if you want.
Okay, just 1 minute.
335 emails.
335 emails for whom?
You.
I'm a busy person, you know.
Wow. So popular.
Don't tell this thing to anybody. A top secret.
Okay.
Yes. So I have shared with you the picture. So the same thing actually. So technically, Tia, the thing is, whatever I wanted to share with you, I have already taught it. So that picture is now, I think it is irrelevant. Okay.
Okay.
Wait, I have shown it. It is loading. Okay. Here, PI and this one. Look here. So I will be just using this part now.
Oh, I is like the regulatory gene.
Yes, regulatory gene. P is promoters. Okay, so this repressor. It come to the operator. Same thing actually.
Yeah.
And then Lac Z. It's the same thing. Lac A, B, C. So these are actually enzyme 1, enzyme 2, enzyme 3. Okay. So these 3 enzymes. So what is operon? And operon is a cluster of genes of a functional unit of DNA which contains clusters of genes which do one particular function.
Okay.
Got my point? So these 3, this is the gene, yes, Lac Z. So this is a cluster of gene. They actually transcribed, first they are transcribed into messenger RNA, and then messenger RNA makes 3 different enzymes, and those enzymes, they control the breakdown of lactose. So
Okay.
these cluster of genes, they are controlling one specific function.
Okay.
You got it?
Yeah.
So we are done with this topic.
Wow. Thank you so much, sir.
So I hope now you can easily go through with the thing and whatever, and